Now we're going to examine some other types of mutations. So one is looking at how does our mutation affect the wild type, genotype, and phenotype. So this picture should look very familiar to you. So when we talk about wild type, we're talking about what is the prevalent or majority of the genotype or phenotype in a population. So here, if we're looking at these penguins, it's going to be the ones that have the black and the white. Then we have one here that's all white. He's considered the mutant, okay? So a mutant allele is going to be the one that's not so abundant, the rare allele in a population. And then remember, polymorphisms is when there's two prevalent alleles or genotype phenotypes in a population. Then we also have something called a reverse mutation, also called a reversion, when a change in the mutant allele goes back to being like the wild type allele, okay? So wild type, polymorphism, mutant allele, reversion. Another way we can describe it is how does it affect the ability to survive? So when we look at a neutral mutation, and we've talked about this in two examples already in this chapter, but these are mutations that have no noticeable effect on a protein's function. Then you have deleterious, where it's going to have a negative effect. And that negative effect can be from being something small that might slightly reduce survival, all the way up into being a lethal mutation that we know is going to cause a premature death. Then we have beneficial. These tend to confer some kind of advantage to our organism or cell. Now, these three here, neutral, deleterious, and beneficial, it can be dependent on circumstances. So if you look back at sickle cell, if you're homozygous recessive for sickle cell, you have sickle cell disease. If you're normal, you're homozygous dominant, but you can get malaria. Whereas if you are a het now this is more beneficial than either of the two. Okay? So where these fall on survival can be dependent on the condition. Then we have conditional mutations, where you can have a mutation, but you might not show that mutated phenotype depending on the conditions. So one well-known example is looking at temperatures. So if we look at fly wings, we can say that they have the mutation for vestigial wings. And if they are developing at a time when the temperature is 25 to 30 degrees Celsius, then they're going to develop this vestigial wing. If the temperature happens to be outside of that, so if it's 15 degrees or 40 degrees, it's suppressed and they will be normal. Okay, so even though they have that genotype, they don't show the phenotype because it's conditional. We have loss of function mutations. So these tend to mess up whatever the function is. So here we're looking at cystic fibrosis. This is a channel protein. Its job is to allow this chloride molecules to go from one side to the other. Well, if you mutate it and it has a loss of function, it can't do its job. It no longer functions as this channel protein, and our ions can't cross. These tend to be recessive. You can have a gain of function. These tend to be dominant. Now we're talking about you are turning on an enzyme or protein or something when or where it shouldn't be, or it takes on a different function. So you're expressing something when you shouldn't express it, where you shouldn't express it, or it has a different function. Then we have suppressor mutations. So this is acquiring two mutations that where one causes a mutant and then the second mutation causes it to be somehow normal again. So here, if we look at our wild type and we notice that this is a complex, in order for it to work, these are two different proteins. We have the square that fits into the perfectly square shaped receptor, so to speak. So we get a mutation, right? And it changes the shape of one of our proteins. Now this protein can no longer fit in the complex that it formed when it was active. But say you get a mutation in that second one and it changes the shape. Now it fits again. So this is the idea of a suppressor, right? So we're talking about somehow changing two things so perfectly that they work again. So there are two ways this can happen. The mutations can be intragenic, so both occur in the same gene. So what happens here is When we don't have a mutation, this thing is working. You get one mutation, it doesn't work. You get two mutations in the same thing, it does work. So in a way, it's almost like one masks the other, or one somehow corrects the other one. Then they can fall in different genes, and it can vary on how they work. So one is a redundant pathway. We have two proteins that carry out the same function. So we're mutating one, and we're stopping it from working, and somehow we mutate another one that is able to carry out the function of the first one, okay? So protein A has a job, protein B has a job. Well, we mutate protein A, it can't carry out its job, but then we mutate protein B so that it resembles the job of protein A, and now it's restored. We can have them work in a common pathway. So two proteins work in the same pathway. You mutate one, it messes up the pathway. You mutate both of them in a way that one corrects for the other, you restore the pathway. So think back to the one where we're talking about the two things that couldn't fit. If that's a pathway, you change it in a way that, well, it works again. You can have them where they work in a complex. So you acquire a mutation in one, they don't fit. You acquire a mutation in both, they fit again. You can have it where it's in a transcription factor. So your transcription factor has a mutation, but then another gene can be mutated that compensates for the loss of that transcription factor. So maybe it's two transcription factors that might be similar, like almost provides like a redundant function. So these are different ways that you acquire genes or mutations in two different genes that somehow interact with each other, and you can restore it to the wild type phenotype. You have position effect. We talked about this when we talked about chromosomal mutations, specifically translocations and inversions. So even though they weren't changing the amount of DNA material, they could change how it was expressed depending on where they fall. So if we look here, and we have this A and B, if we move A in a way that is up here by B, and now let's say this is an enhancer, this regulatory sequence, and we move A up there close to it, we're going to hop over the insulator if it has one. Now that regulatory sequence can control both A and B. So whereas here A didn't have an enhancer, here it does. Now we could be ramping up how we're expressing this, and that's going to be a problem because it's not supposed to be expressed as much. We can also take it, and here we have a euchromatin and heterochromatin. If we translocate an active gene from euchromatin into heterochromatin, now it's inactive. So remember that euchromatin tends to be transcriptionally active, whereas hetero is silent. Well, if you put this active gene on hetero, you're not going to make it anymore. So now we're messing up how much of that gene we're supposed to have. So far, so good. Then we have these broad classes that I alluded to at the beginning of another video, where they can be somatic or germline. And we know somatic cells occur in the body cells that don't give rise to gametes, whereas germlines give rise to gametes. So if you have a mutation in a somatic cell, you are putting a mutation in just the daughter cells of that cell. Remember mosaicism? We're back at this again. So if you have a mutation in a somatic cell, it undergoes mitosis. All resulting cells from that one cell are going to continue to have that mutation over and over again. So here we are, we have several different cells. Only the cell affected and the cells that come from it are going to be mutated. Everything else is going to be normal. Now we are mosaics. We have cells that have different DNA in them. For these, it can be as simple as there's no phenotype. Whatever is going on, we don't have a phenotype. It's not enough. Maybe it's in a gene or something that's not needed in this tissue. Or maybe it's not enough to have a phenotype, so we have enough normal to still be normal. Or maybe it's detrimental. That cell dies, no harm, no foul. Or maybe it's so unlucky that it causes cancer. Okay. Now, if it's a germline mutation, now it's different because now we're going to mutate that egg or that sperm. And now when they come together, every cell after them has it. So if you mutate this sperm, sperm meets egg, that sperm's half is coming with the mutation. So every cell from the zygote on has that mutation because it's from the germline. So if it's a germline mutation, it affects the entire organism. Whereas if it's a somatic mutation, it's just a part of that organism. And it's not likely to be passed on, whereas germline mutations can be passed on. Okay? So if you affect the germline, you affect an area. If you affect the somatic cell, you affect an area. If you affect the germline, you affect the entire organism. We have this idea of the random nature of mutations. So they did some research. They proposed this random mutation theory. All it's saying is mutations are random. Okay? Mutations appear very randomly. Well, like theories, these things, after we find, we get more and more sophisticated with our experiments, we can do different things, we can alter these. So we're going to alter this one because some genes are more susceptible than others to a mutation. And why is that? Okay? So it can be size. The bigger a gene is, the more likely it is to occur mutations. The next one can be located. So where is that gene located on a chromosome? It could be in an area that is just more susceptible to mutations. And then some genes have hot spots. So there are certain regions of a gene that are more likely to mutate than other regions. Okay? And this is just depending on what that gene is. And think back to like if it was a trinucleotide repeat, those are areas that are going to be more likely to be mutated. Okay? Questions, comments, concerns?